Good evening. I'm Kelsey King, the Bahnick Fellow in Evolution, Ecology, and Organismal Biology, and in January and February I was at Iowa State University in Ames, Iowa. And this background here is actually sphagnum, which is the moss that I was working with. I worked with peat soil, so just kind of a little side note as to what this background is. It is photo edited, so it's a little better for the slides. I was in the Hofmockel lab in Bessie Hall. It was very cold and covered in snow when I worked there, but I was in her lab working under Dr. Ashley Kaiser, who is a postdoc there, and she actually has her own status to be a primary investigator, so it's kind of like her first start working as a supervisor. And I was working with the Spruce Project, and that's in soil microbial ecology, which is just the community of living things in the soil. And this project is actually funded by the Department of Energy and the Minnesota government. And it's at this Marcel experimental forest, which I did not get to go to, but I got the samples from there. And this forest actually has these platforms that allow you to sample in the bogs. And the bogs are basically just floating carpets on water, is kind of how you can think about them. And if you're walking on them all the time, it's going to be disturbing that. So we have these platforms to avoid us disturbing all of the soil. And these little circles are just the sampling sites, so we kind of have those designated. And this site is actually going to eventually simulate climate change to see what's happening in these bogs with the future warming temperatures. And I think the highest is like raising it by 12 degrees and the lowest is by 1 degree Celsius. So my project goals specifically were to learn about bogs and climate change. So bogs have large stores of carbon. It's especially because it's so frozen and so cold that a lot of the time the microbes aren't decomposing anything, but the plants can still make a lot of stuff during the summer, and it gets stored over the winter. And the reason why this is a problem is that with climate change, we're going to have warmer summers, so we could potentially be releasing more carbon, and it's going to be released as carbon dioxide, which would be a positive feedback to climate change. So that's kind of why we have all of these high-level government agencies concerned about this, because the United States does have quite a few large bogs, especially in Minnesota. And I was just conducting a pilot experiment to help characterize this soil community. It's creating these microcosms here. And so I folded up, and I say folded because it's, again, just decomposed moss, which you can see here. It's really hard to work with, but I folded it up, put it in these vials, and gave the soil different increases in various nutrients. And that kind of simulates different changes that we might have happening in the future experiments. We want to know what is happening in this soil before we start raising the temperatures. It's kind of just characterizing what do these microbes like to eat. And finally, I'm an environmental studies major, and I do have to write a capstone project, and this was my capstone project. So I hope to start writing that paper, because I knew once the block plan started up again for me, it was not going to be as much of a priority. So the methods, I had 63 of those vials, which are 2 trays and another small tray of those. And it was 5 different substrates, which is just the kind of the food I was giving the microbes. And it was at 4 concentrations for each. And it was just sampling how much carbon they produce in response to this. And we simulated summer temperatures and the conditions at which the carbon is mostly stored, so at saturation below the water level. You can see me here sampling. We have the gas sample right there, and I had to sample with a gas-tight syringe, kind of catch my breath every time I say that now. 63 times with flushing the needle in between with air, so that's just like pumping it a bunch. And needless to say, it was a very long, arduous process, but I got through it and it was really fun. And it ended up with some cool results. So here I just have the graphs. This is cumulative carbon dioxide that's just over the entire 45-day experiment. And so we have chitin, which is just part of fungi, and there's a lot of fungi growing in these bogs, so we would expect the microbes to be used to fungi components. Glycine, which is an amino acid, so it's necessary for protein, so hopefully all microbes want that. Glucose, which is a sugar, again, hopefully all microbes want that as well. But then we also have 2 organic acids, which are excreted by plant and fungi a lot, especially for fungi when they're decomposing things, and plants typically throughout their whole life. And then on each of these graphs, we have a water treatment, which is just a 0. So in every single graph, you'll see that there just for reference. And you can see that we didn't get a lot of difference from the water. And that's really important to note because we're trying to get difference from the water. We want to see how they're reacting to these additional nutrients. And so some of those outcomes were we figured out that we needed to have higher concentrations as the input. So they're not reacting to this. So at the concentrations we added, yay, no positive feedback to climate change, which is a good note. Or we needed to kind of pulse the substrates, because if we're trying to simulate, increases by plants, like those organic acids, they're going to be doing that continuously. So if we wanted to simulate a more natural condition, we'd be pulsing it. So adding it, weekly or every other day or something like that. And this is me at my workstation. I was there for the majority of my internship in that chair, reading a ton of papers and setting up all of the spreadsheets we used, working on my capstone, which I did finish my methods and introduction section while I was there, which was great. And I've recently turned it into my chair, so yay! That's a really good outcome, even though the experiment wasn't the results that we were expecting. So lessons learned, pilot experiments are where you mess up. These methods that we were using were mostly untested by people, especially people interested in soil ecology. They typically work with forest soils because those have been, more interesting. And you can actually pour those soils, fun fact. Made me a little regretful on what soil I worked with. Double checking calculations is really important, especially if they're not yours, just to make sure that everything's working right. And speaking up if you have ideas is really important, especially if you have ideas that are going to make your life more easy and more efficient. It's good to speak up and let them know. I learned how to use this gas syringe. I did get blisters pretty much every time I sampled. It's a very, very rough process, but necessary. And it's actually a really fun skill. So if I ever have an experiment where I want to see how much carbon dioxide or water is being produced, I can do that. And I learned a lot about graduate labs. I had the opportunity to visit multiple graduate labs while I was there, and that was a really enlightening experience for me. I got to meet people from all sorts of disciplines and all ages and different experiences, and it was a really great time for me. It let me kind of figure out what I wanted to do and what things I wanted to look for when I'm looking for graduate labs. More on those impacts, specifically for my academics. I decided that I want to work in desert ecology, which seems kind of different from what I worked on, but it's great because I didn't have the motivation to really get into that, and being around so many graduate lab people and with the primary investigators and the postdocs and PhD and master students, it was very enlightening for me, and so I do have a lot of motivation to actually get into those projects now. And soil ecology is, part of my scientific thinking. It's not something that I've really gotten into in my classes, so it's nice because that's a really important part, especially when you're looking at plants. And networking, I realized that it actually is really important for sciences, which I didn't really think about before. So using that is going to be a really great way for me to, get those references and also just meet really fun people who can let me know of cool opportunities that they've had in the past. So for future impacts, having graduate lab experience is really great, especially when it's combined with all of the field and lab work I've done in Cornell in the past. And that is going to make me a lot more competitive. I think it was really the one thing I was lacking. And like I said, I learned to speak up and have confidence in my abilities, which is really important, just being able to assert myself as a scientist and recognize that my ideas are valid when I'm talking to the primary investigators and things like that. It did test some of my limits, especially professionally, just like talking with people that I had no idea who they were, never met them before, and like nobody knew them because it was at a very large school. And usually I knew somebody who knew, oh yeah, I know this person in the English department already, even though that's not my department. So it was really enlightening and, helped me figure out ways to talk to people a lot more professionally than I do here. And I'm not scared to live alone anymore, which is great. So the thank you slide. I am so thankful to my donors. Without this, money from them, I wouldn't have been able to do this opportunity, and it's been so great for me and let me do some really cool research. My site supervisor, Dr. Ashley Kaiser, who funded all the research side, and also, just watched me and made sure I knew what I was doing. And my faculty sponsor, who's my academic chair for environmental studies, and actually pushed me to do this. So thank you to Craig. And everybody in the Hofmockel lab was really great too. So shout out to them and everybody else who supported me during and before the internship, especially the CCEC. And special thanks to my mom who came here tonight, and she's the one who moved me in and out of so many places over the last 4 years. So props to her. It's been a rough ride for her, but she's been really supportive, which was great. So, and thank you all for coming tonight.